Hello guys, today our topic is apoptosis. In this lecture we will discuss about the causes of apoptosis, introduction to apoptosis and what are those enzyme and factors that involve in the apoptosis. First, the to what is apoptosis? Apoptosis is genetically programmed cell death. Genetically programmed cell death. Genetically programmed cell death. Unlike necrosis, in apoptosis there will be no protein degeneration. No protein degeneration. Number second, there will be no leakage of enzyme. No leakage of enzyme because the plasma membrane inside the in the process of apoptosis remain intact. There are certain important points that we should know about the apoptosis. That number first, it may be it may be physiological. Number second, it may be pathological. Remember that necrosis will be always pathological, and unlike necrosis, apoptosis may be physiological or pathological. Number second, apoptosis occur in single or few cells. In single or few cells. Necrosis does not occur in single cell. Number 3. Inflammation is absent in apoptosis. Inflammation is absent because the cell membrane remain intact, because the plasma membrane or cell membrane remain intact, and due to that the intracellular enzyme will not come out of the cell and will not release into the tissue. So what is mean by physiological apoptosis? In which conditions physiological apoptosis will occur? In which condition there will be physiological apoptosis? If we look embryologically, we know that in organogenesis in organogenesis number first, cell in between the digits cell in between the digits undergoes apoptosis. In between the digits the cell undergo apoptosis. Number second, esophagus. It is just like during embryologically esophagus is just like rod, and during organogenesis in between cells in the esophagus undergoes apoptosis. For example, this is esophagus and it is like a rod, solid rod, and during embryogenesis or organogenesis the cell undergo apoptosis and it becomes open. There is also occur apoptosis in other situation. For example, woman breast during lactation. Breast during lactation. We know during the lactation the level of prolactin hormone, the level of prolactin hormone is increases. But when women stop when women stop lactating, when women stop lactating, level of prolactin decreases. So the cells that are dependent on the prolactin, they undergo apoptosis. Those cells that are dependent on the prolactin hormone during the lactation, they undergo apoptosis. Number fourth, during the menstrual cycle. During the menstrual cycle, just before the menstrual bleeding, just before the menstrual bleeding, estrogen, progesterone level decreases. Estrogen and progesterone level decreases. And we know that endometrial cell are dependent on the progesterone level during the menstrual cycle. So when progesterone level decreases, endometrial cells undergoes apoptosis. So these are the physiological condition in which cell undergoes apoptosis. Number 5, we all know about that. During immune system development of immune system development, we know that T cells go to thymus for maturation and also develop TCR, T cell receptor for antigen. T cell go to thymus for maturation to develop TCR for the antigen. But sometime T cell make such a receptor that sometime T cell make a receptor that recognize their own antigen of the body. So these T cell undergo apoptosis. So this was physiological condition in which the apoptosis occur. Number second, what are those pathological condition in which apoptosis is occur? Pathological conditions. This is occur when there is damage to DNA. For example, when there is damage to DNA, for example, due to radiations, toxins, etc., or chemotherapeutic agents. Number second, this will occur when there is accumulation of misfolded protein. Accumulation of misfolded proteins. We had already discussed in previous lecture about the misfolded protein, that it brings the concept of endoplasmic reticulum stress. When protein is formed, it goes to the endoplasmic reticulum for the proper folding. But when there is a misfolded protein accumulate inside the endoplasmic reticulum, there will be risk to develop, because there will be more number of misfolded protein will form inside the cell, and there will be risk to develop cancer. What will be the morphological features of cell in apoptosis? Morphological features. Remember, morphological features are most important for the exam question. Number first, there will be shrinkage. Cell will be cell shrinkage. And remember that this is the earlier feature. This is the earlier feature of apoptosis. Earlier feature of apoptosis. Number second, chromatin will be condensed. Chromatin will condense inside the cell. And remember, this is the most common feature of apoptosis. This is the most common feature of apoptosis. Number third one, we had already discussed that during the process of apoptosis, plasma membrane remain intact. Plasma membrane remain intact. Remain intact. Number 4, inflammation is absent. Inflammation is absent during the process of apoptosis. Why inflammation is absent? Because the plasma membrane is intact and the intracellular enzyme will not leak out into the tissue. Number 5, apoptotic bodies are formed. Apoptotic bodies are formed. What are apoptotic bodies? These are small bags. These are small bags in which cellular material are packed. Are packed. Cellular material are packed. The apoptotic bodies are removed by the macrophages. So, what are the mechanisms of apoptosis? Remember, there are 3 phases in the process of apoptosis. Mechanism of apoptosis. 3 processes are involved in the process of apoptosis. Number first, initiation phase. Initiation phase. Number second, execution phase. And number third is removal of dead cells. Removal of dead cells. These three phases are involved in the process of apoptosis. What are the enzymes that involve in the process of apoptosis? Enzymes. There are 2 enzymes involved in the process of apoptosis. Number first is caspases and number second is endonuclease. Endonuclease. These 2 enzymes are involved in the process of apoptosis. What are caspases? What are caspases? These are the family of proteolytic enzymes. This is the family of proteolytic enzymes which have cysteine end, which have cysteine amino acid in active pocket. In active pocket and cleave the target peptide and cleave the target peptide at aspartatic aspartatic site. What is mean by this? These are the proteolytic family. These are of the proteolytic family members. For example, this is caspase. For example, this is caspase, and it has amino acid called cysteine. And for example, this is a protein inside the cell which have different protein, and I just guessed that at this point this aspartate amino acid is present. Aspartate amino acid is present. So these caspase enzyme cleave the peptides at those points where the aspartate amino acid is present. That's why this enzyme called caspase. That is why this enzyme called caspase. Caspases. C means cysteine, aspartate, and proteolytic enzyme. Proteolytic. That's why it is called caspases enzyme. Remember, caspases are of two types. Caspases are of two types. Number first is initiator caspases. Initiator caspases, and number second one is executioner caspases. These initiator caspases are help in initiation phase of apoptosis. These are, for MCQ's point of view, remember that these are of caspase at 9, 10 and 8. These are the initiation caspases. And number second one is executioner caspases. Executioner caspases. These are those caspases, executioner caspases. Caspases. These are those caspases that help in executioner phase. That help in executioner phase of apoptosis. Remember that these are caspase 3, 6 and 7. What are those factors that involve in the process of apoptosis? Factors. What are those factors that involve in the process of apoptosis? These are of two types. Number first, pro-apoptotic factors. These are those factors that stimulate the process of apoptosis. Stimulate apoptosis. That stimulate the process of apoptosis. Remember that these are two factors. One is Beck's, and second one is Beck. Beck's and Beck are the stimulator of apoptosis. Number second, anti-apoptotic factors. These are those factors that inhibit the process of apoptosis. Inhibit the process of apoptosis. The process of apoptosis. These are these are number first BCL2, number second BCLX2, and number third is MCL1. So this was all about the intro of apoptosis, and inshallah in the next video we will discuss the mechanism of the apoptosis.